now we are coming to the third topic, which is development.
Okay.
Development in plants. Okay, so what are the stages of development, and what structural differences it plant undergoes, we are going to see here. So development is nothing but all changes in life cycle from germination of the seed to senescence to the old, to when it becomes old. So this time period, this lifespan of plants are known as development. How it is development? Okay. So here, with response these development stages, with response to environment, they change their structures of plant and everything. It adapts to the environmental conditions. Okay. So with response to environment or phases of life, these form different kinds of structures. Okay. So these different kinds of structures, the capability to form different kinds of structure by a plant is known as plasticity.
Plasticity, okay.
Okay. Example, heterophyly in cotton, coriander and larkspur. So in these three plants, they show plasticity. Okay. And if you take buttercup, no, they have different kinds of leaves in air and different kinds of leaf in water. So this is also a kind of heterophyly, which is plasticity. Okay. So if you see in larkspur, earlier when it is in juvenile phase, when it is starting to grow, the leaf of these plants will be like this. Structure will be like this. When it becomes adult, the leaves will change the structure like that one. Okay. Whereas in buttercup, the terrestrial leaves which are present in the air conditions, the leaves which are present in the air conditions, these leaves appear like this. They appear like this in structure.
Okay.
So when the same buttercup, when it is present in water, these leaves are present in water, the leaves will be looking like that like that structure.
This is present in water, or this is not?
No, that is larkspur. That is early and adult stage. Here it is terrestrial and water conditions. Okay. So this kind of changes in plants, whether it is leaves, branches, or anything, it is known as plasticity of plants. Okay. So here it is also the factors affecting these development is also light, temperature, and everything. What we have seen in the early condition. Here we saw the factors affecting the growth in plants. So the same factors here also, it affects these structures in plants. Okay. So next we are going to see the fourth topic, which is plant growth regulators. Okay. We are going to see about the fourth topic, which is plant growth regulators.
Okay.
So plant growth regulators are also known as PGRs. Okay. And these are also known as plant hormones, phytohormones and everything. So here, there are many plant growth regulators which are produced in plants. Okay. So which are these plant growth regulators are small, simple molecules and are chemical compounds which have many chemical composition. Okay. So first we are going to see about indole compounds. What are the examples? So indole compounds, indole 3-acetic acid is an example of indole compounds. Okay. This IAA, indole 3-acetic acid. Okay. So and then adenine derivatives, it is made from adenine. Okay. So these are N6-furfuryl aminochlorine, which is also known as kinetin. Okay. And then derived from carotenoid. Carotenoid derivatives are
Abscisic acid.
abscisic acid. Okay. And then the third one is, fourth one is terpenes. So these terpenes are gibberellic acids are made up of terpenes.
So G, gibberellic acid, and 3.
GA3.
Okay. Gases.
And these hormones are gaseous form. Okay. So gases, ethylene, C2H4. Okay. So these are generally plant growth regulators which are found in plants. So some plant growth regulators have promoting function. Promoting function like it promotes seed growth, it promotes germination, it promotes senescence and everything. And some plant growth regulators have inhibiting function. It does not. It inhibits the growth of seed, it inhibits growth of plants and everything. And some fit for both. They also promote and they also inhibit. Okay. So here those which are growth promoting hormones regulators are auxins, gibberellins and cytokinins. Okay.
Promoting cytokinase. Okay. Promoting.
And the promoting is promoting. So promoting is auxins, gibberellins and cytokinins, okay? And these are generally known as auxins. Indole 3-acetic acid, which is also known as, these compounds, single compounds are generally known as auxins. And gibberellins are this one, gibberellins. And cytokinins are this one, chitokinins. So these compounds are cytokinins, okay? And growth inhibiting is abscisic acid. It inhibits growth, abscisic acid. And then these fit for both are, it is fitting for both promoting and inhibiting. So it is a kind, but it is largely inhibiting, okay? But also it promotes. So example of this one is ethylene, which is a gaseous plant hormone, okay? So now we will see the discovery of the plant growth regulators, okay? So under plant growth regulators, we are going to see our second subtopic, which is discovery of plant growth regulators, okay? So here we are going to see two discoveries of plant growth regulators, okay? So all the five plant growth regulators, all the five hormones were discovered accidentally, okay? And in first Charles Darwin, okay, he paved the way for the discovery of the first hormone. So Charles Darwin and his son Francis Darwin, so they observed coleotides of canary grass. Coleotides are nothing but the tip of the grass, okay, the tip of the plant. So they found that it was growing towards light source. It was growing towards, if sunlight is here, it was going towards here. If sunlight is here, it is going towards here, okay?
Okay.
So this kind of mechanism is known as phototropism, okay, growing towards light. So they found that tip of the coleoptile, only the tip of the coleoptile caused a blending of the coleoptile, okay. So the tip of the coleoptile was the main reason for this phototropism, okay. So here they just found this one, but they did not identify the hormone, okay. So later auxins were identified, isolated by F.W. Went. He only first isolated auxins from the tip of the coleoptiles of oat seedlings, oat plants, okay. From oat plants he discovered these auxins. These auxins are nothing but indole 3-acetic acid, and these are indole derivatives, okay. So these were first found by Charles Darwin and his friend Francis Darwin, and it was isolated by F.W. Went, okay. So next comes the discovery of gibberellic acid, okay. So here Bacon, okay, Bacon, it is also known as foolish seedling, okay. So Bacon is a disease, okay, it is a disease of rice seedling. In rice seedling, these Bacon disease occur often, okay. So it was caused by fungi Gibberella fuscori, okay, and it was found by Kurosawa. This Kurosawa did, he took uninfected rice seedling, okay, and he applied sterile Gibberella, this fungi Gibberella fuscori, and he found the disease Bacon, okay. So it is caused by gibberellic acid, okay. So next we will see the other three discoveries of hormones, okay. So now we are going to see some more discoveries of plant growth regulators. So already we have seen two discovery of plant growth regulators, which were gibberellic acid and auxins. So here we are going to see some more plant growth regulators. There are totally 5, right? So we have seen 2. So remaining 3 we are going to see. So later F. Skoog and Miller, they found cytokinesis promoting active substances, okay. So what is cytokinesis? Cytokinesis is nothing but after the division of nucleus, which is karyokinesis, there occurs cytokinesis, okay. After the division of nucleus, cytoplasm also separates. So separation of cytoplasm is known as cytokinesis. The separation of nucleus is known as karyokinesis. So first nucleus divides and then the cytoplasm divides, okay. So So this cytokinesis promoting active substances, they discovered these active substances and named it as kinetin. So kinetin is a derivative of cytokinins. Okay, cytokinins is also known as kinetin. So later, in mid 1960s, okay, in the middle period of 1960s, three researchers made separately, led to the discovery of inhibitor B, abscission 2 and dormant. Okay. So later they proved that all these three discoveries were chemically identical, all were same, and named it as abscisic acid.
Okay.
Okay, ABA. So we have seen 4 discoveries. So the last one is, and after that cousins. So cousins were scientists. So they observed some release of
Whose cousins?
Cousins is a name of a scientist.
Yeah.
Yeah.
Okay.
So what did he observe means he observed that the release of volatile substance from ripened oranges. So from ripened oranges there were some release of volatile substances. Volatile substances means some evaporative substances. Okay. So they found that this substances which is evaporating from the ripened orange, it can fasten the ripening of unripened bananas. So the volatile substance which is coming out from ripened oranges can fastly ripen the unripened bananas.
Okay.
So after that they discovered that volatile substance and named it as ethylene. So it is a gaseous plant growth regulator. Okay. So up till now we have seen the discoveries of all the 5 plant growth regulators, all the 5 auxins, gibberellins, cytokinins, ethylene and everything. So next we will be seeing the physiological effects of plant growth regulators. Okay.
Fifth topic.
No, subtopic.
Okay.
Okay. Physiological effects. What each plant growth regulator is doing. What are the activities of them will be studying separately. Okay. Now we are going to see the last subtopic under plant growth regulators, which is physiological effects of plant growth regulators. So here we are first going to see about auxins. Okay. So auxins are plant growth regulators, or also known as hormones. So these were first isolated from human urine. Okay. And auxins is a general term used for all indole compounds, indole derivatives, that is indole acetic acid and other natural and synthetic compounds of indole derivatives, and which are having some growth regulating properties.
Okay.
So altogether it is known as auxins. It is a term used for these compounds. So these auxins are produced by growing apices of stems and roots, like coleoptiles of plants. Okay. So indole 3 acetic acid is isolated from plants. So therefore it is a natural auxin.
Okay.
And NAA, naphthalene acetic acid, and 2,4 dihydroxy acetic acid. So these two are synthetic auxins. Okay. And so these auxins initiate rootings at stem cuttings. So if you cut a stem and if you insert auxins in them, they produce roots from the stem cuttings. Okay. So these initiate rootings at stem cuttings. And these auxins promotes flowering in plants. Example, in pineapples, if you place these auxins, it starts to flower. Okay. And these auxins prevent fruit and leaf drop at early stage. Okay. At early stage, the fruit and leaf drop will be prevented by these auxins. And it promotes abscission of older parts. Abscission is nothing but the weathering. Okay. It promotes abscission of older parts like decaying leaves and everything. So it promotes the weathering of that parts, but it prevents the falling of early stage parts in plants. Okay. So here apical dominance is nothing but in most plants the growing apical part. So the growing apical bud inhibits the growth of lateral buds. Okay. It inhibits the growth of lateral buds. So when you cut the apical bud, directly the lateral buds will grow. Okay. which is found in tea plants. So therefore in tea plants always they will be cutting like this to prevent apical dominance. So it will with the lateral sides of the plant will grow.
Not grow height.
Yes.
Grow width. Okay.
So that is why they are cutting tea plants. It is known as hedge making. Okay. And these auxins induce parthenocarpy in tomatoes. Parthenocarpy is nothing but production of fruits without fertilization. Okay, that we have already seen in the chapter. Okay. So production of fruits without the fertilization. Without fertilization occurs, they will produce fruits. So it induces parthenocarpy in tomatoes. Okay, in tomatoes no fertilization occurs when if you add auxin to the plant. And these auxins help in the cell division, differentiation and everything. So this 2,4-dichlorophenolic acid, it is widely used to kill dicot weeds. Okay, it does not kill monocot, it kills dicot weeds. Okay. So this is about auxins. So next we will see the second plant growth regulator, which is gibberellins. We will be seeing about gibberellins. Okay.